I, Dr. Muhammad Akram, uh, Associate Professor, Department of Eastern Medicine, Government College, University, FAISALABAD. I will discuss about gastric juice. It is secreted from three types of cells in the stomach. Chief cells secrete pepsinogen, parietal or axonal cells secrete ICL and intrinsic factor, mucous cells secrete mucin. Composition of gastric juice includes water, inorganic and organic matter. Inorganic constituents include HCl, most important, sodium, potassium, and phosphate, very less. Organic constituents, mucin, pepsin, gastric lipase, traces of other enzymes, and intrinsic factors. Gastric juice is highly acidic. Its pH ranges from 1.3 to 2.6. Amount of gastric juice secretion is about 2 to 3 liter per day. Properties and functions of gastric juice. It is secreted pepsin. It is secreted in the zymogen inactive form called pepsinogen, which is at first activated by ICL and then by pepsin itself. Pepsin is a powerful proteolytic enzyme. It is an endopeptidase, therefore, it hydrolyzes the central peptide bond of protein. It curdles milk. Gastric lipase. Gastric lipase breaks down short and medium chain fatty acids. It is destroyed by trypsin. Hydrochloric acid. It activates pepsinogen to pepsin by removing the amino acid covering the active form of pepsinogen, maintains the acidic pH of the gastric juice so that pepsin can function efficiently for the digestion of protein. It has antimicrobial activity. It helps in the absorption of iron by converting it from ferric to ferrous form. Mucin. It acts as a lubricating agent. It forms a slippery layer over the gastric mucus. This slippery layer acts as a barrier against the action of gastric juice on gastric wall and it prevents it from autodigestion. Mucin secretion is increased by irritant of gastric mucosa, hyperacidity, and vaginal stimulation. Intrinsic factor. It is protein in nature. It is essentially required for the absorption of vitamin B12. Deficiency of intrinsic factor results in deficiency of vitamin B12, which eventually causes megaloblastic anemia. Since the vitamin B12 deficiency is due to the lack of intrinsic factor, it is called pernicious anemia. Regulation of gastric juice secretion. Secretion of gastric juice continues under all circumstances. Secretin Secretion is caused by small amount of gastromucosal, gastrointestinal hormone released from pyloric antrum by vagal stimulation. Secretion of gastric juice in response to meal can be divided into three phases. Neural or cephalic phase. Neural sensory stimulation is caused by sight, smell, and taste of food, and it increases gastric secretions. The vasovagal reflex also causes secretion of gastric juice. It is caused by the distension of stomach, especially the fundus, stimulating stretch receptors in the stomach wall. The stretched receptors stimulate vagus nerve, resulting in the release of acetylcholine in the vicinity of gastric glands, causing secretion of gastric juice. The juice secreted in response to vagal stimulation is very rich in HCl, pepsin, and mucin. Gastric phase starts when food enters the stomach and stimulates the secretion of the gastric juice. The effect is through local hormone. Effect is more profound in response to what soluble products of protein digestion, peptides, and amino acids and alcohol. These products cause release of a hormone called gastrin from these cells, endo enterochromaffin cells of pyloric antrum. Gastrin enters the blood, reaches gastric glands, and stimulates parietal and chief cells to secrete a juice rich in pepsin and HCl. Intestinal phase occurs when chyme, partly digested food from a stomach, enters first part of duodenum. Digested lipids, proteins, and low pH of chyme causes secretion of three gastrointestinal hormones: secretin, gastric inhibitory peptides, cholecystokinin, pancreazyme, Pancreazymin is the chyme which the with these three hormones inhibits gastric phase and initiates intestinal phase of digestion. Secretin causes pancreas to secrete pancreatic juice rich in bicarbonate. It also inhibits the secretion of HCl by inhibiting parietal cells of the stomach. GIP, along with secretin, inhibits HCl secretion by the stomach. CCKPZ is the predominant hormones controlling this phase. It stimulates the gallbladder to contract and release the hepatic and pancreatic enzymes stored in it into the duodenum. This phase represents only 10% of the gastric secretions. Pancreatic juice. Now we will discuss about the pancreatic juice. It is clear solution containing water, inorganic and organic matter. Inorganic constituents includes mainly bicarbonate, chloride, sodium, and small amount of phosphate, calcium, and magnesium. Organic constituents include mainly enzymes which are described here. Pancreatic juice is alkaline, pH 8 to 8.3 due to high content of bicarbonate. Amount of pancreatic juice secretion is 1.2 to 1.4 liter per day. Enzymes of the pancreatic juice, trypsin. It is secreted in the zymogen form called trypsinogen, which is converted to the form called trypsin by the enzyme enterokinase and then by trypsin itself autocatalysis. It is an endopeptidase, therefore it hydrolyzes the central peptide linkage of protein. It activates chymotrypsinogen to chymotrypsin. It accelerates blood clotting. Chymotrypsin. It is secreted in the zymogen form called chymotrypsinogen and is then converted to the active form chymotrypsin by trypsin. It is an endopeptidase, therefore it hydrolyzes the central peptide linkage of proteins. Carboxypolypeptidase, it is an endopeptidase and hydrolyzes the terminal peptide linkage of protein at the carboxyl end. Elastase is specifically hydrolyzes elastin but can hydrolyze other proteins as well. Collagenase, it specifically hydrolyzes collagen. Pancreatic lipase hydrolyzes triglycerides, acts only in the presence of cell, bile salts. Pancreatic amylase, it is identical to salivary amylase and converts starch, glycogen, and dextrin into maltose. Deoxyribonuclease causes hydrolysis of DNA. Ribonuclease causes hydrolysis of RNA. Phospholipase A2 causes the hydrolysis of teflon and lecithin. Cholesterol ester hydrolyzes cholesterol esters to cholesterols and fatty acids. Regulation of the pancreatic juice secretion regulated by neurogenic and hormonal mechanism. Neurogenic mechanism regulations, parasympathetic stimulation of pancreas results in enzyme-rich secretion of pancreatic juice. Sympathetic stimulation of pancreas resulting in the inhibition of the secretion. Vagotomy inhibits secretions. Stretching of fundus of the stomach results in increased secretion of the pancreatic juice. Hormonal regulation. Secretin. It is gastrointestinal hormone which is secreted from the duodenum and the upper part of the small intestine. It is released in response to HCl, which is present in food coming from the stomach. The pancreatic juice secreted in response to secretin is watery, more alkaline, but deficient in enzymes. Secretin relaxes the lower part of the esophageal sphincter, decreases the activity of the pyloric antrum, increases bicarbonate sec- secretion in bile, increases secretion of duodenal glands, and increases motility of the small intestine. Cholecystokinin, pancreazymin. This gastrointestinal hormone is secreted from the upper part of the small intestine and duodenum. It is released bile in response to proteins and fatty acids present in food. The pancreatic juice secreted in response to this hormone is rich in enzymes. CCK, PK, PZ causes contraction of gallbladder. It destroyed gastric motility. Now, we discuss the bile. Before discussing the bile, I am closing this session. Thank you very much. जाके दबा